Hi students, welcome to another episode of Microbiology Decoded. So today we are going to study about Taenia Saginata and Taenia Solium. So both are flatworms coming under the phylum Platyhelminthes and the class Cestodes. So let's get into the class. So as you can see in this picture, we can see Taenia saginata worm and Taenia solium cousin worm. So its adult form, it is around 5 meters in length, which reside inside our intestine. So this is their picture, and they both are known as beef tapeworm or pork tapeworm. This Taenia saginata is a beef tapeworm and Taenia solium is the pork tapeworm, and it causes some infection like teniasis and cysticercosis. And next is the source of infection, from where we are getting, how we are getting the infection. So from cow or any other cattle, this Taenia saginata will infection occur. If we are eating raw or uncooked beef, we will get the Taenia saginata infection. And from the pork, if you are eating uncooked or raw pork, we will get the Taenia solium infection. So next is the mode of infection, how this infection will transmit. So by eating undercooked or raw meat or beef. So if you are eating the beef, it contains larvae known as Cysticercosis bovis larvae. When we are eating our undercooked pork, it contains Cysticercosis larvae, which is known as Cysticercosis vellosiae, and it causes intestinal stenosis. So coming to the infective form, the larval stage called cysticercus is the infective form. There are two types of cysticercus, as we said already, that is the Cysticercosis vellosiae and Cysticercosis bovis. So next is the clinical picture. So as this adult worm are very long, about 1 to 5 meters long, they are likely to cause the abdominal pain and vomiting due to this intestinal obstruction. So the patient gives history of passing small pieces of tape-like structure about 1 to 2 centimeter long in their stool. Sometimes they can see that these are crawling in the freshly passed stool, and they are known as the proglottids. And coming to the lab diagnosis, the sample we are going to take is the stool sample, and here we can see the egg of Taenia. In the stool sample, we have to search for the egg of Taenia. So as you can see, it is yellow color because we are using a bile stain. So the bile stain gives this egg a yellowish or sometimes brown color. So this egg is around 30 to 40 μm in their size, and it is a spherical shape. They are having 3 pair of hooklets. So we can see here they are having a hooklets. Visible hooklets are inside, around 6 hooklets. So these hooklets help to anchor in our intestinal mucosa. They have to be attached in our intestine and causes infection. So the hooklets are meant for anchoring, and they have an oncosphere. Oncosphere is the developmental form of the larvae, and they are having a thick radiated shell also. And the Taenia solium eggs are infective to man and as well as the animals. So it is the diagram of the egg. As I said already, this is having a developmental larval form, that is the oncosphere, and hooks. They have hooks inside that to anchor in the intestinal mucosa, and they are having an enterophore. This enterophore will protect the oncosphere from any harsh environment. So it is acting as a protective cover in total. So let's move to the life cycle. So next we'll see about the life cycle. So here it's begin. The Taenia saginata and Taenia solium egg or gravid proglottids in feces and passes into the environment. Proglottids which contain 500 to 800 eggs, and it contains both female and male reproductive system in it. When this egg or gravid proglottids will come into the environment, this cattle and or pig will ingest it, in the vegetation. While they are having their food, this gravid proclotis or egg will go into their intestinal tract, and the egg hatches, and this oncosphere will come out and they penetrate into their intestinal wall and circulate into the musculature, and this oncosphere develop into cysticerci in their muscle, and when the human ingests this containing meat, they will get the infection. So Taenia Saginata, Taenia Solium, Solex attached to our large intestine, and if it's developed into an adult, it will attach into the small intestine and through the phases of the human, and it will continue as a life cycle. So this is the life cycle of both Taenia Solium and Taenia Saginata. Next, we will see about the cysticercosis. So what is cysticercosis? So as we know that teniasis is acquired by the injection of larvae, while cysticercosis is acquired by the ingestion of egg, which are often passed by the same individual. So that is known as the auto-infection. If you are having the larvae that is which will cause to the teniasis, and if you are ingesting the egg and which causes the infection, it's linked to cysticercosis, and sometimes this often pass to the same individual and it causes the auto-infection. So we'll see about the clinical features. The clinical features which involve the subcutaneous nodule formation. We can see a nodule which formed in our subcutaneous tissue, which is under the dermis. So if the nodule developed inside a vital organ like brain, if it's developed in our brain, the patient may have convulsions. So the patient will develop convulsion. That is a symptom which our body movement is very irregular, and that is the same symptoms as we studied in the rabies virus. So now we are going to see about the difference between Taenia solium and Taenia saginata. We said already Taenia solium is a pot tapeworm and Taenia saginata is a weed tapeworm. So they can present for many years in our small intestine, and the definitive host of Taenia solium is man and rarely dogs. Taenia saginata is only man. Intermediate host is pig and man in Taenia solium and cattle in Taenia saginata, and intermediate stage in solium is Cysticercosis cellulosae and Taenia saginata is Cysticercosis bovis. And this side we can see this infection and a disease in man. Sometimes the Taenia solium which causes the auto-infection, but Taenia saginata will not cause any auto-infection. So as I said already, we can do the bystaining for identifying their egg, but also we can do some radio diagnosis. Like this, we can see in the brain, we can find like Dottinke appearance or rice grain appearance, and here we can see, and this is the brain radio diagnosis, and this is the whole body radio diagnosis. So here and all, we can see the rice grain appearance. So, if this class helped you understand the difference clearly, please like, share and subscribe to Microbiology Decoded for more simplified microbiology lessons. In the next class, we will discuss about something interesting topic. So, thank you.